There is a question in the Q&A here. Can you comment on how you would design a characterization release panel for these EVLPs or VLPs, especially for regulatory submission? Any high-level comments you have for that? Yeah.
So,
while we're on the topic. What, Gloria?
Gloria is actually in clinic, so she can probably give you a lot more details. We're early. We have not actually initiated any regulatory conversations for our EVLP platform technology. That said, I think it's going to look a lot like any sort of characterization release panel that you have for lentivirus, because our particles in particular do look a lot like a lentiviral particle. And I think there's going to be, increased scrutiny on, for example, host cell DNA, host cell protein contamination, things like that. But I think it's pretty standard if you're familiar with lentiviral release criteria.
Gloria's nodding. Do you have anything to add, Gloria?
No, I'm just agreeing with her. I think it's probably it's a very tough question, and I appreciate the audience participant for asking it, to know what's important in it. And there's been a pretty recent guidance. I mean, one of the good things of the COVID pandemic, I guess you could say is good, is that the FDA got really pushed to go fast, right? And we haven't had guidances on some things since 1996. We're still using the old guidances, but the guidances that are out on things like genome editing and LNPs, et cetera, they came out fast, right? Because they had to, and they got a lot of information very quickly. And so there's been some uncharacteristically new guidance that gives you a lot more information about what's actually important to look at. So I think, I guess I would guide the audience participant who asked the question to look into those FDA guidances and they're kind of in they put them all in the cell and gene therapy, but on gene editing, and you do get quite a bit of information about what kind of things are important. Also, when you don't know what to do, you find out what somebody else did that did it. And, there have been some things that have been approved recently. Like I know that Vertex has a drug, Casgevy, I guess, for
sickle cell.
Sickle cell. And there have been, a couple of other that are close. And so there is a precedent out there for what kinds of things need to be reported out in these characterization, release, and stability type panels. So it's worth taking a look.
Yeah, I think so if I'm hearing you both correctly, right, there are ways that we can kind of infer, right? It's not necessarily for an EVLP, but we can take the learnings from viral delivery, et cetera, right? Some of the other advanced therapy approaches to help build our own panels.
And that's what the agencies do, right? They go with what they know and they extrapolate from there and then figure out how close they are and how well that works, right? So
talk to the agencies.
And they're not compendial generally. So this is a lot of work, right? These aren't compendial assays typically because people haven't been doing them before. Some are, but some of like the potency and identity assays, they're sort of de novo and need to be qualified and eventually validated.
Yeah. Well, Gloria, since you're big on my screen, do you want to jump in and share some of the what are some of the benefits of working with extracellular vesicles compared to, say, an LNP, right? Where does this delivery vehicle shine in terms of its scientific properties as well as its clinical or manufacturability?
Sure. At first, I guess I'd say they're very different for application. Like, I'm not sure you would go after the same application with and pit the two against each other.
Okay.
So what we go after is really regenerative healing. And so it's not typically a single silver bullet that says, oh, you're changing this thing or you're delivering this oligo. Now, some people do, and that's okay. But for example, epidermolysis bullosa is a monogenic disease. It's a COL7 deficiency or absence. And yet that's not all there is to the disease. So a company recently developed, Crystal just recently had approved a gene therapy for a COL7 gene therapy for epidermolysis bullosa wounds. And it does have some reasonably good efficacy. But there are other things that it can't treat, and that is the immune dysregulation that the patients have, those sorts of things that are underlying some of the healing process, such that the wound doesn't just necessarily close, but it's also more skin-like. So it's some of the best skin that they have on their body when they've been treated with this. It heals up and it's it's better than most of the rest of their skin, and the healing seems to be very durable. So that's what we're talking about, the regenerative piece. Now, if you're going after something that you just need to deliver a specific RNA for a specific purpose, I'm not sure I would pick it, an EV or an exosome for that. I think the exosome is more a milieu of things. Even if you add and engineer and add an RNA, which is difficult, it often breaks them, and they don't. That's a tough. But people do it, and companies have done it, and they're working on that. So that's one way to do it. But it's probably a easier path to go with an LNP if you just want to deliver, or some of these other technologies, if you're just delivering a specific RNA versus a milieu of cargo from a stem cell, for example.